So thank you very much, Joanne, and ILSI IFBiC. Thank you very much for the opportunity to come and meet many of my friends from around the world, from India, from China, from South America, from Europe, and a few from the U.S. as well. And also meet new people who hopefully will still be a friend in the end. No, I'm sure that, you know. Anyway, I want to cover a lot of material that is hard when you're the only person talking about a subject that's kind of complicated to suddenly jump in and have 25 minutes, 30 minutes to talk about it. So please forgive me. And if afterwards you want some additional material, please email me and I will send it to you. You also have, I think, the slides online. I think that it is important for me to make this statement. When I joined Monsanto in 1997, I was asked to come and help predict whether a protein, a novel protein, could be an allergen, and I kind of laughed and thought there's no way. You know, I'd been working in immunology and so forth. But really, that's not necessarily the biggest risk. The biggest risk, if you're allergic, is do you know what you're eating? Does it contain your allergen or not? And for the direct safety assessment of the new trait, the new biotech product, the question is really, did you transfer an allergen into a new food material that you wouldn't expect? When I first joined, they'd already set up essentially the safety assessment, and it was kind of set in 1992 under the federal FDA had put something on the Federal Register. And it also included the idea that if you made a transgenic soybean or wheat, which are pretty common allergens, maybe you should look at endogenous allergens. Did you increase the endogenous allergens or not? And to me, that sounded pretty reasonable because a fair number of people are allergic to soybeans and wheat, not to corn and rice. Very few people have food allergies to corn and rice. It sounded reasonable, and for a number of years I really thought, you know, you really have to do that. Well, people have gone on and said now you should do the assessment in corn and rice, at least in some countries, and I scratch my head and I say, why? Where's the real risk, and how do you evaluate it? And more I looked at it, and I work with Steve Taylor in the Food Allergy Research and Resource Program. We work with food companies where the real task is to make sure if you made a cookie that contained peanuts and then you make a cookie that does not contain peanuts, that you don't carry over peanut into the unlabeled non-peanut containing cookie. That's the real risk. And the same is true for GM. That's the real risk. I'm listing here things that some people would say it's important to say where you get money. I work Allergen Online is sponsored by the 6 major biotech companies. We get money from them. They don't get a vote in the allergens that are included in the database, but they want a safety assessment tool that is pretty universal. We've had 2 EPA grants that worked at IgE binding. We also have one now on animal models and allergenicity that I co-share with Joe Baumert. I'm going to present some data that was sponsored by and to look at products by BASF and Bayer. And there's another new product that I'm not going to tell you the developer of, but it's also going to be included a little bit. I want to mention that I had 5 years work previously standardizing allergenic extracts at Hollister Steer, and that's a very similar process to what we're talking about today. If you make a diagnostic extract for evaluating whether or not somebody's allergic to peanut, is it uniform and how potent is it? You have to do that. And so it's directly related. I've done endogenous allergenicity assessments or directed studies in a GM wheat product. I want to talk about non-GM maize because, and we used animal antibodies there to look at lipid transfer protein. I'll present some data. We also have looked at 5 GM soybeans. One of those was at Monsanto, and the other ones have been with other companies since then. There are different methods that you could use to evaluate whether or not you have changed the endogenous allergens in a food-grade material. And the first study I did was with 1D immunoblots and individual sera for that, and then ELISA inhibition with pooled sera. And then we've also done 2 studies with individual sera with 1D immunoblots under reducing and non-reducing conditions, and also 2D immunoblots with individual sera at the request of EFSA, the European Food Safety Authority. We also used ELISA inhibition with pooled sera for those 2 products. And then we've also done one where we also did direct binding with individual sera with ELISA, as well as 1D and 2D blots. I would note that things have changed over time. You already heard that Roundup Ready soybean was evaluated for endogenous allergenicity. That was with pooled sera looking at 5 different soybeans, I think, or at least there was 5 different subjects in it. There was no clear IgE binding difference, and that product is, what, 80% of the market of soybeans, I think, in the U.S. or something like that. I don't think that there's any indication that that's caused a problem for people. I would just remind you that the focus of the safety assessment really primarily is on the new gene, the new protein. Did you really make a problem that would impact the health of humans or animals? And also, both for GM and non-GM materials, the real risk isn't the GM. The real risk primarily is it peanut, is it soybean, is it cow's milk? If you have a food product that contains wheat, well, that has to be avoided by people with celiac disease. Roughly 1% of the world has celiac disease and has to avoid eating wheat, barley, and rye. If you have an allergy to peanuts, you have to avoid peanuts. If you have an allergy to cow's milk, you have to avoid cow's milk. We also know that if you have many of the legumes we eat in this world have to be cooked in order to inactivate lectins and proteases and amylase inhibitors, things like that. So clearly food itself is not necessarily safe for everybody. It's how you prepare it, and it's whether you're allergic or have celiac disease. This is the overall safety assessment under Codex, and under most countries have similar regimes. The real risk for food allergy is the source of the gene, taken from a product or from an organism that has allergies, and if so, is the gene expressing a protein that's an allergen? Also, is the protein so similar expressed from that gene that it would likely cause cross reactions in those allergic? If the protein is from wheat, barley, rye, or very close grass relative, is it one of the glutens that causes celiac disease? Those are pretty easy to assess. Can be expensive, and you sometimes get surprised, but those are pretty easy, and those are the big risks. The next risk is, could a new protein that's never been known to be an allergen suddenly sensitize people and become a major food allergen? That's harder to assess. It's less certain. The risk is fairly low, but we have some measures. It's also a little less sure in the answer. And finally, here's the question. Did insertion of the new gene significantly increase the endogenous allergens? And I'll remind you that in Codex it says of food materials that are known to be commonly allergenic, which in what we know today says in peanuts, in wheat, and in soybeans. It doesn't say in rice. It doesn't say in corn. Those are not known to be commonly allergenic. And that's under the compositional analysis. I would remind you that in terms of the safety assessment, what's most important is that we protect people who have allergies or have celiac disease, and those people have to do a lot of things in order to remain safe. And it requires appropriate diagnosis, it requires appropriate labeling, and in most countries today there are important labeling laws for the major allergens. It requires food companies and commodity crop people to be careful in their controls. It requires accurate labeling on menus and packaged foods, education of restaurant workers, and in terms of GM developers, it requires them to do those first 3 things I talked about to help make sure you're not introducing a major allergen or a celiac disease agent into something like rice, where somebody wouldn't expect to have a problem. If you look at the real world of allergy, and I'm just showing a few pictures of a few people who have food allergies, and if you give them the food, they can react very quickly. To understand what it is that caused these reactions, you need a careful clinical history. You need often an elimination diet. You may need skin prick tests. You may need food challenges. This is one of the better ways to tell, but clinical history is the most important. You may use in vitro IgE binding, but there are many false positives with IgE binding, some false negatives. There are many false positives with skin prick testing, some false negatives. If we look at the real world of food allergy and risk, this is from a paper, Sisher and Sansom 2010, and you look at young children versus adults. Milk allergy is the most common thing. Most kids outgrow their allergy. Eggs is also outgrown by most people. Peanut isn't outgrown by very many people. Tree nuts also isn't outgrown. Fish, oh, well, young kids rarely eat fish. Young kids almost never eat shellfish. Those are major allergens. So adults have commonly the allergens. Wheat and soy, roughly 0.4%, 0.3%, depends on the world area. In Japan, maybe adults 0.2% instead of 0.3%. Sesame seed is eaten in some parts of the world much more commonly. Total allergies. Okay, these are my educated guesses. Corn or maize allergy is less than 0.05% of the population. Rice, 0.001 to 0.005%. They're not major food allergens. If you test with raw materials, you test by skin prick test, you test by IgE binding, I would say at least 10 of you will show up as likely to be allergic to rice or corn. That's wrong, though. That's not going to work. Probably nobody is allergic to cooked rice if they sit in this room, but maybe somebody. If you have an allergy, what happens? Well, you have antibodies, IgE antibodies, that sit on basophils and mast cells on Fc epsilon receptors, and if you encounter the protein that is bound by those, and if there's two receptors, you can induce this cell to release histamine and leukotrienes, but actually it takes 10 to 100 of these events on one cell at one time to really cause that cell to release. It takes tens to hundreds of those cells to cause the symptoms that can happen very quickly. Well, how can we say how much of a change in an endogenous allergen is too much? Because that's an important question. If we're going to ask this, it's kind of like how much iron is too much? How much lipid is too much for me to eat? How much sugar? I don't know. But allergens, it's much more of a strict thing. How much of an allergen, though, does it take before you have a change? Now remember that if you're allergic, you should avoid the food you're allergic to. Well, we might use some of the threshold studies that have been done as a guide to determine how much is too much. Steve Taylor in the Food Allergy Research and Resource Program has worked with a number of people, and I'm going to describe in a minute just a little bit of the studies. For the major allergens like peanut and milk and a little bit for soybean, there's some data that we know about how much. And you might be surprised at the range of variation from allergic person to allergic person. It's important to realize as one of these evidences that in the challenge doses that the best doctors in the world do, they take 2 to 3-fold increases in each step of a challenge. So here I am in the test room. I get the first dose. It starts at 3 milligrams. 30 minutes later, I didn't react. I get the next dose. It's 6 milligrams. 30 minutes later, I didn't react, and it's up to, 12 milligrams on up to 4 grams or more. So that gives you some indication that if it's not at least a 3-fold difference, the best clinical allergist in the world is not too worried that he's going to kill you or have a serious reaction at the doubling to tripling step. So that's one set of things to think about. So the doses that were done in the peanut allergen studies that were done in France, 276 people, there are very few people who will give you an objective symptom. That means if I'm looking at you and giving you this stuff, I can see you react. It's not, I feel funny, it's I can see something going on. You get a hive, you get a fat lip. That's an objective symptom. So down in the range of 3 milligrams, a few people do react. Some people react at gram quantities, 1 gram, 3 grams. So in the population of people that are allergic, there's that kind of variance. Most of these people will not react. If they react with an objective symptom, they will not suddenly have systemic anaphylaxis when you double or triple or even quadruple or quintuple the dose. A few might, but that is very rare. So this is one big important study, and that's really for food companies to say, how clean does my food line have to be in order that I'm not worried when I make the next cookie that doesn't have peanut? Here's a study from Germany, well-respected allergists looking at milk, egg, wheat, and soybean, looking at different doses that range from 3 milligrams to 3,000, 5 to 4,000, 3 to 2,000 milligrams. So huge doses, and they're doubling, tripling in every dose stage. They looked at quite a few different subjects with different reported allergies. This is the percent of subjects that reacted at a given dose. Clearly, there is a dose-response effect. With soybean, wow, you don't get very many people reacting until you're at 3 grams. So that's quite a dose. If you look at the severity of reaction, which they also looked at, and the darker the color for egg and milk and soy and wheat, the darker the color, the more serious the reaction. So clearly some people get a serious reaction at a fairly low dose, but it's not generally until you get to a high dose before you get some severe reactions. Most of these people in wheat will react very quickly, but a few react, 6 hours after the first dose. And that's a scary factor with soybean and wheat as well. Some people react instantly, some are very slow, and we don't totally understand it. Most people react to peanut very rapidly. But let's get to the real world of eating food. So we're not eating soybeans, we're not eating wheat grains, we're eating food. All of these foods have soybean of different doses. And when you think about what variety was in this versus this versus this versus this, we don't control that in the commodity chain. So how much variation is there today? If we're going to talk about safety and health, we need to think about those issues. And it's also true that not all allergens are equal as a source of allergy. Not all proteins are equal as allergens. If we look at peanuts, and there's good data, and I don't have the references listed, but I can get them. Peanuts probably kill roughly 50 people in the U.S. per year. Could be as low as 20, could be as high as 100. We don't know. They're not coded that clearly in the hospital usually. There are 3 major allergens, the 2S globulins, RH2 and RH6, the viscilins, RH1, lysins, RH3. Those are major allergens, major seed storage proteins. There are a number of minor allergens, including in peanut. L2P is not very important in peanut, maybe because of those. Profilin, BET-V1, those are generally mild allergens out of 10,000 genes and hundreds of proteins of different types in the seed. Soybeans, probably less than 1 fatal reaction in the U.S. per year. 3 moderate allergens, beta-conglycinins and the glycinins, also maybe P34, which is also GlyMBD30KD. A number of minor allergens, 20,000 genes. Wheat, no documented cases of fatalities globally, and I just got on email and talked to 8 of the world's best food allergy experts last week to find out for sure. And what about food allergy risks? And that's what they said. So that's an important thing. There are 1 to 3 major or moderate allergens. Often people do react a little bit late, and so it's kind of hard to look at in a challenge test. Maize or corn, no published reports that I'm aware of a fatal reaction globally. 1 major allergen, which is lipid transfer protein, 4 to 5 minor allergens. However, you can find IgE binding to a number of proteins. So as I said, we have allergen online database. This is the last release. It's updated yearly, and it's got some 1,600 different sequences of different allergens. Some of them are isoforms. A number of them really just have a little bit of IgE binding. They're not absolutely proven. We don't take 1 protein and dose you, if you're peanut allergic, and say, is this the protein that's causing you to react? We don't do that. Instead, we find out peanut caused your allergies, and then we look for IgE binding and things like that, and we say this is a presumed allergen. A number of proteins in here are pretty well documented. A number of them in here really just bind IgE and are unlikely to cause reactions. Keep that in mind. We also have a new trial celiac database on there that if you're taking a gene from wheat, barley, or rye, you might want to use that. And there's a description of how you might use it. I'm also the secretary of the IUIS Allergen Nomenclature Committee, which is another database information source. So assessing endogenous allergens. What could go wrong? How could you change endogenous allergens? Well, you might have an increase in the expression or the accumulation. That seems like the most logical thing. You also might have point mutations that change the allergenicity. You might have a gene rearrangement. You might have a duplication. All of those things can kind of work together to change endogenous allergens. You also may add or subtract a glycosylation site, an asparagine-linked glycosylation site, but the glycans are almost always shown to be false positives. That is, you can get IgE binding, but they will not cause a reaction. There is an exception to that, and that's an animal glycan, not a plant. There is some minor evidence that, or some evidence that glycans in some plant proteins may cause minor reactions. Other post-translational modifications that can occur are probably not too important in terms of allergenicity, but we could argue about that. So very little is known about natural variation. The concentration of the allergenic proteins across current non-GM subjects are not really well known. You know, we could think a lot about the things you've heard in the last 2 days, right? There's a huge amount of genetic variation, and it's different between corn and soybean varieties and wheat. Wheat can be diploid, tetraploid, hexaploid. So you could imagine there's a tremendous variance in the allergens expressed in those plant grains. Those are important things. You also have environmental factors that will modify the expression of genes. You also have breeding to improve crops that will change things. And we also have what? Food processing. I'm in food science. After you grow this crop, you make it into a food that somebody can eat. And that can impact the allergenic proteins and how they're presented. It's important to realize each species has multiple possible allergens and isoforms, minor variants, and you can look and you can do IgE binding, but sometimes IgE binding doesn't mean that the protein is an allergen. So I turned this diagram that Laura presented a little bit differently. I want us to think about, you know what, after we make a transgenic plant this way, and we've done a careful job, we've taken a gene from a non-allergenic source, we've inserted it into soybeans, what are we going to do with it? We get somebody to approve it, and then we say, okay, now we're going to do this, right? Because now we need to have it in different elite lines and things like that. So it's important to realize that once you get the GM product, you're going to then breed it into different backgrounds. And sometimes it's going around the world. It goes to India, and they use very different germplasm there. They have to, or it won't grow. And so now you've got some change in what you had versus the study you just did. Should you reassess it? Oh, gosh, I hope not. I don't think you should. I don't think the risk is there. But you have to realize that after many back processes and things, the endogenous allergen level may be very different than what you tested for 4 years before. Okay, so what methods do you use to look at this stuff? And I would say that every single method I'm listing here is imperfect. You will get a lot of the data you need from many of those methods, but oh gosh, I'm in trouble. But you won't get the full answer in any case. Usually you use immunoblots or RAST inhibition. You can also use animal antibodies, and I'll show you an example. I'm not going to talk about proteomics because they're complicated, and I don't think we know enough about all the allergens to know anything about whether or not we found them. You also have to realize you have to extract the proteins from the source material, and different proteins have different solubilities, and they will behave differently. You may see it differently than you think. Here's a thing with, oh, these are fruits. These are not GM, but this is apple lipid transfer protein, peach lipid transfer protein, probably major allergens. There's a 100-fold difference across varieties. There's a 39-fold difference across varieties. Natural variation. Here we did a study with a graduate student who looked at 9 non-GM hybrid varieties grown in 2 locations in Nebraska under irrigated and not irrigated conditions. Here's one of the irrigated sites, 4-fold difference in LTP binding, the major allergen in corn. Oh, well, you go to the non-irrigated site, and then you see that there's up to 15-fold difference. And, whoa, that's a lot. That is a lot. Think about that. Okay, we can have different IgE binding sites on a protein. You can have conformational epitopes, which means if you can denature this protein, you no longer bind IgE. You can have sequential epitopes that it doesn't matter if it's boiled, although if you don't denature it, this may be hidden. You can have CCD or carbohydrate binding, and again, that's almost never important biologically. You can use different methods, and I'm not going to spend a lot of detail, but you can use reducing, denaturing, non-reducing, denaturing. You can use native protein, and you can use either direct binding or you can use inhibition binding to look at the binding levels and see how much IgE do you get. Serum testing, if you have to get people with the right serum allergy and right amount of IgE. And remember that the IgE binding alone does not equal allergy. That's an important thing. You also can get misleading results because there are antibodies against IgE that you use as a secondary detection system that will detect IgG. All of us have IgG antibodies against some of our dietary proteins, so that's very important. This is, oh gosh, this is ugly, right? Stain gel, peanut, kidney bean, chickpea, blah, blah, blah. This is IgE binding from one subject who has allergies to navy bean and to milk and soybean, peanut, a number of allergies. What we do know from inhibition is this is all CCD binding and doesn't seem to cause reactions in this person. Here's a case where here in the kidney bean, this is probably not CCD binding. It's probably IgE binding to the protein. That could well be what causes their allergy to navy bean. Here's peanut. It's not CCD. You cannot inhibit it, and I'm not going to show you the methods that we used. We've done soy IgE endogenous allergenicity tests, and it's based really on diagnostic regulations. If you're going to make a diagnostic antigen, what do you do to test, and how much difference do you need? Well, for diagnostic tests, 50% to 200% is okay for a standardized allergenic extract. If you're going to do serum testing, you need to have subjects. You need to have clinical people who can diagnose the subjects. You need to have IgE binding data. You need to know something about their symptoms. This is one of the soy studies. This is BASF, and on these gel blots under reducing and non-reducing conditions blotted with one of the subjects allergic to soybean, you can see different banding patterns between this subject and this subject. Here's what's on there, isoline, transgenic, and then commercial line 1, 2, 3. So what we do is we actually can find some funny things going on, sometimes in a 1D gel, sometimes in a 2D gel. This is a 2D gel, stain gel from isogenic, transgenic, and 2 commercial lines. You don't see much difference. I'm not giving you time to look. Sorry. If you looked at this and you did densitometry and you picked spots and you looked at the protein, you could try and figure out, is it glycinin, is it beta-conglycinin, whatever. But it's pretty tough and you don't see any real big differences. Now you do IgE binding on a blot and you see a very different picture, and if you try and match the big black spots with the stained protein spots, you'll be surprised because most of those are fairly non-abundant proteins. And what's the difference? Isoline, transgenic, oh, this commercial line has 2 spots that do not show up in these other 3. Is this protein, is this soybean more allergenic than the other 3? I don't think we have proof of that. We have proof that there's a difference. We don't have proof, is this going to cause harm to that subject? This is a different subject, not as well labeled, but I just wanted to show you that some people have IgE that binds to just a few proteins. Oh no, here's the parental line. It's only got 1 spot here, not 2. Does that mean it's less allergenic? No, you don't know that. That's also grown in one location, So you don't know if that represents every place these genotypes will grow. If you do ELISA inhibition, and we did, and we used replicates, 3 replicates of this assay, and we used a standard Dakota plates, and we inhibited with standard in each of the lines, and you look at the 50% inhibition values, and we did not see any statistically significant difference. So that study is now submitted to a number of agencies. Bayer FG72 just showing 1 serum, 1 subject, reducing conditions, non-reducing conditions. Here's probably an important allergen for this subject. I just want you to notice that here's a lot of IgE binding. We don't know what it is. We don't know what it is. I think it's a glycan, but we're not sure. That's a non-GM line. Does that line represent a big difference in allergy? I don't think so. When we do an inhibition study, that particular line falls off very quickly. Apparently, the affinity of IgE binding to that particular soybean extract is much less, and as you increase the dose, the IgE starts changing and falls off. Does that represent less risk? I don't think so. I don't think we know that. Here is another product with a number of transgenic and non-transgenic lines. Here's 2 subjects under reducing and non-reducing, and the 4 and 5. 4 represents the non-isogen control. 5 is the soybean, the GM. Here's some other subjects, and you'll notice, oh gosh, there's a difference here. Well, which one is worse off? Is it the 3 commercial lines that have that extra band, or is it the non-GM and the isoline and the GM? We don't know that. This is direct ELISA, individual subjects. So here we have all the individual subjects. Here we have commercial lines and the GM and isogen that are going at 3 different geographical locations. And you can see there's a lot of diversity in total IgE binding for any one subject. They're not huge. This is optical density readings. It's not absolute IgE, but you see differences. And if you tried to do a statistical comparison of 1 GM, 1 isoline, and which location you pick, you could have a statistically different result or not. Is there a hazard that's different? I don't think so. As somebody who works in food allergy, I don't think so. When you run the statistics on this, this pink line represents no difference between the isogen and the GM. There's no statistically significant difference. If you look at locations, it's not statistically significant, but there are probably some differences. If you look at 4 of the 5 commercial non-GM lines, there are statistically significant differences, increases in IgE binding across all subjects and locations compared to the isogen. Does that mean that they're a lot more allergenic and we should take them off the market? I don't think so. Thank you. I'm almost done. So anyway, here's kind of the wrap-up. I've looked at results of our studies that we've done when I was at Monsanto, at BASF, at Bayer, and another company. We see some differences. I don't think they're important. If we focus on any difference being a risk, then we might say, oh gosh, not this one's okay, not that one. I don't think that's important. And especially since we have to say, you know what, if you're allergic, you should avoid the food. The people at risk should avoid soybeans. It doesn't matter if it's GM or not GM. In summary, the safety assessment of GM organisms has been proven. The stuff I haven't talked about, evaluating the protein from the transgene. It's been proven to be very good at weeding out allergenic proteins. We do know that we can evaluate endogenous allergenicity, but the results are not absolutely clear-cut. Most importantly, though, there's not a risk, because if you're allergic to soybean or maize, you should avoid those foods that contain those materials. If you're not allergic, you should eat them because they're good nutritional products. There's no proof of increased risk from increased allergen expression, and we do not have established thresholds that say this much is too much, this much is acceptable. We're looking at differences we don't know that matter, the problem. There is an exception. I say don't do these tests for the common transgenic products. If you claim you have developed a hypoallergenic peanut or soybean or wheat, then you better do these tests and a lot more, because otherwise, if you label this hypoallergenic and somebody with peanut allergy eats it, you may kill them. So you've got to think about that. It's also important that there's no way to have zero risk. So we have to accept some amount of risk. And one could claim, well, what if you increase the allergens 5-fold? Would there be a risk? You can't know that, but guess what? If I eat this candy bar versus that one, that has 5 times more soybean in it than this one. What's the difference? So there, I gave you a very biased talk. I'm sorry. But that's after 15 years of experience. Here are my graduate students who have done most of the work. And you'll notice from India, from China, from Sri Lanka. I have some I have one U.S. student and a few from Africa and so forth. Anyway, they're very good students. I work with Steve Taylor, who started FARP, and Joe Baumert is also there. There are other people, postdocs and a technician. Margita Worm from Germany supplied some of the soy subjects. Barbara Balmer-Weber supplied some of the soy subjects. Very good food allergy experts. I've had money from EPA, from FARP, from mostly food companies, from a couple of the biotech companies, and Allergen Online is sponsored by the 6 biotech companies.